Good morning, good afternoon. Hi, everybody. It's really exciting to see people filtering in. I've got a bunch of people joining us live, and I know that a lot of people will be watching the recording. So we're thrilled today that we have Madeleine and Chris, who volunteered when I do a lot of reaching out to people and asking them to do things. And so they were generous enough to agree to give an overview today of different emerging genetic treatments for SCN8A, and give I've been through the slides, some great overview to help us kind of understand how they work and what they are. And then also to delve a little bit into the experience that they are just starting on a path down, which is looking at an ASO. The N of 1, N is kind of like, the number in the study, and 1. So it's just a treatment for one person with this group called nLorem that is just emerging as looking at treating genetic epilepsies in just one patient and getting a sense of if these ASOs can work and how they work. So there's a lot of really exciting stuff happening right now. And basically, I'm going to pass it over to Madeleine and Chris in just a second, but I'll say that as we're going through, please feel free to use the chat to put in your questions or, things that come up for you. If you say, oh yeah, that's interesting, I heard about this, or I want to know more about this. And then we'll spend some time at the end discussing and having a chance to ask more questions, because I know this is a topic that we're all really interested in and excited about, but have a lot of questions about. So I'm going to hand it over to you all. Thank you so much for agreeing to share both your scientific experience and your personal experience as parents with us.
Yeah, we're happy to be here. So just in terms of background, so I'm Madeleine Oudin. I'm the Tampo Family Assistant Professor of Biomedical Engineering at Tufts University. And so I actually do have a PhD in neuroscience, but then I moved to cancer research. And so my research in my lab is focused on breast cancer, metastasis, and resistance to drugs. But we've recently been studying the role of ion channels in cancer, mostly focusing on potassium channels, and also studying how nerves contribute to cancer. So I've kind of come back into the neuroscience space. And so, yeah.
So I'm Chris Burge. I'm a professor of biology at MIT. My background is in computational biology, and my lab at MIT studies gene expression, RNA biology, computational biology, and genomics. Recently, we're interested in the role of alternative splicing in different diseases, including SCN8A.
So this is our daughter, Margot. She's 18 months old. She has 2 de novo mutations in SCN8A. Started having seizures at 3 months old, mostly infantile spasms, and then more recently tonic and clonic seizures. And so she's been on 9 AEDs, currently on 3, and on the keto diet. Spasms are controlled right now, but she's still having daily tonic seizures. She's very developmentally delayed, very low tone, has no head control, has CVI, and, but is working hard in lots of therapies and just got a gait trainer recently that we're experimenting with. So that's been, so we do want to have a disclaimer, right, that we are not clinicians. We're not giving any medical advice. We're just scientists who are using our knowledge to explain how some of these existing and emerging therapies work and, what options are available. We don't know everything that's happening, in all the pharmaceutical companies, things like that. So, we're just kind of giving an overview of, how these things work, what's kind of the things that are the closest to new treatments and what are some that are, kind of more further away. So in terms of therapeutic options, we have to go back to this central dogma, which is what drives biology and how our cells work, right? So we have our DNA, which is, the information that is unique to each individual and that drives the generation of proteins that are the effectors in the cells and in our body. The proteins are what, get things done and have us functioning as human beings. And so the DNA sequence dictates what protein is made, and the in-between molecule that is made to go from DNA to protein is called mRNA. And so this kind of going from DNA to mRNA to protein is what's called the central dogma of biology. And again, it's what dictates, the function of our cells and who we are as individuals. And so it's important to note that in the context of SCN8A, and you may have heard this and it's confusing, that the gene is called SCN8A, but the protein that is made from the gene is called NAV1.6. And so, sorry, that's a typo. It's NAV1.6. So it can be a little confusing, but when we talk about the protein, we should be talking about NAV1.6. And when we talk about the DNA, we're talking about SCN8A. So we know that it's mutations within our DNA sequence that our children have that, then impact what protein is made and how it functions. So in terms of treatment options, the most common and what, most of our kids are on are drugs or small molecules, right? So these are compounds that impact protein function. So our common sodium channel blockers like trilaptol or dilantin, they impact the function of the sodium channel. But many of these drugs are not specific, right, to NAV1.6. There are newer drugs like the Neurocrine drug that is more specific for NAV1.6, and the Praxis drug that affects different properties. And so, but either way, these just impact the protein function. They don't touch the DNA or the mRNA. So what you most commonly hear as gene therapy is the idea that you are replacing or changing or adding DNA into cells to make a protein that maybe is no longer being made. So gene therapy really involves, adding or modifying DNA. So you're going kind of back to the source to then, add a DNA sequence or correct an endogenous DNA sequence so that then you can get a correct protein. And then ASOs, which are a newer treatment modality, or antisense oligonucleotides, actually impact the mRNA. So they target this intermediate step. So they're not correcting the DNA in any way, but they're able to be used to change what protein is being made. Okay, so those are just general kind of concepts in terms of these different treatment modalities that we'll talk about and how they impact DNA versus RNA versus protein. So the other thing to keep in mind in terms of these treatments is, how easy they are to make and actually get to the brain, right? So there are kind of three main characteristics that we'll discuss, which is one is how they're actually delivered, right? How they're given to patients. That's really important in terms of, what we're doing clinically. And then there's also, how easy they can be, how these treatment options can get to the brain, right? And then how long it actually stays in the body. And how long it stays in the body is important, both in terms of how often you need to take a given treatment, right? Versus, but also, how quickly it will leave your body if it's not working, right? Sometimes if treatment's not good, we want it to be leaving the body soon so that, it's not like causing any more damage. So in terms of the drugs, that our kids, most of us take, right? Most of these are taken orally. And even though, we are able to, the field has been able to make drugs that can get to the brain. Now, still targeting the brain is not always that easy because there's this protective blood-brain barrier, which is you know, a structure that is there to help protect the brain from other things in our body that might hurt it. So in general, it is harder to get drugs to the brain, but, you know, this is still something that the field has been able to accomplish. And so we can, you know, take these drugs that can get to the brain. A lot of these drugs, you know, within, you know, several hours will then kind of leave the body. And that's why, you know, we need to give our kids these drugs, you know, every couple of hours or so that the level stays high. But it also means that, again, if a drug is not working well, then, you know, within like a day or two, we can be pretty sure that that drug is out of their system very quickly and that it's not there. So, you know, that's also helpful when there's toxic responses. So in terms of the DNA replacing therapies or modifying or addition, like gene therapy, and so one example, and we'll talk a lot about SMA, spinal muscular atrophy, because it's a genetic disease that's had a lot of these treatments approved for it. So, so they have a genetic therapy called Zolgensma, and this is actually given IV. So it's pretty easy to deliver. Because it's much harder to get your DNA to be any DNA that you would give in the body to get it in the brain and expressed and kind of entering all cells in the brain to have a good effect. So that's pretty hard to do. I think there are some clinical trials for other gene therapies where they can inject into the brain. That's pretty new, but that is happening. And the thing to note with these treatments is that it's a one-shot treatment and that's it. Like your DNA, that DNA is in there and, you know, it will be active for a very long time. So this is a great thing because, you know, it's a one-shot treatment and like you're not constantly having to take something. But again, it also means that, you know, if there's an issue with this, like there's nothing we can do about it. And then ASOs are somewhat in the middle in that there is an ASO, and again, this is approved for spinal muscular atrophy, Spinraza, and by a company called Ionis, and they have a way to get it to go to the brain by injecting it into the spinal cord. This is called an intrathecal injection. So this is doable. Obviously, this needs to be done, you know, in the hospital setting to be able to inject it into the spinal cord. And it stays in the body for, you know, months at a time. So Spinraza is given every 3 months to patients. So, you know, it still can leave, you know, the brain over time, but there are, you know, constant injections. So these are just, yeah, kind of things to keep in mind in terms of these therapeutic options. So in terms of SCN8A, how does this come in? So just so we're all on the same page, brain activity is regulated by a combination of cells that are excitatory and that are, you know, activating the brain, and then cells that are inhibiting it. So there's a balance between that activation, the excitation, and the inhibition. And so seizures are generally caused by an excess in brain activity, either because there's too much excitatory input or too little inhibitory input. And so we know that SCN8A is mostly present in those excitatory cells. And so SCN8A is a gene that is present on chromosome 12. And so each of us have 2 copies of the SCN8A gene. One copy we inherit from the mother and one from the father. And so mutations that cause a gain of function in the sequence of SCN8A, you still have 2 functional copies of SCN8A, but one is more active because of this mutation that causes it to gain extra function. And so in this context, you know, for these patients, what we want to try and do is reduce the activity or the amount of SCN8A. Because to kind of bring it back down to normal. And then for loss of function mutations, what we have is that one copy of the gene is not functional. And this means that you only have one functional copy of the gene. And so technically, in terms of, you know, restoring it back to baseline, you'd want to increase the activity or amount of SCN8A at this level. So in terms of therapeutic options, sorry, we're going to explore and talk a bit about kind of how these different options relate to these gain of function and loss of function mutations. Any questions so far?
Madeleine, I would like just to ask, for example, in the case of gain of function, so one copy of the gene is perfectly functioning, and the other one is not working. Is that correct?
So, I mean, it's not, if it's a gain of function mutation, it's still working. It's just working differently. It will be more active. And so it's still working and making a protein, but the protein functions differently. So for many mutations, like the SCN, the protein, the NAV1.6 protein will stay, the channel will stay open longer, so it's more active, or it won't inactivate as quickly. Different mutations have lots of different effects.
Okay, but the other copy that is not marked with the star here in the picture means that this is perfectly
Yes.
functioning.
Yes.
Okay, thank you.
Yeah.
It is difficult for us to know if we are in the case of gain of function or loss of function. Do you know how we can try to figure out for our child if he's gain or of loss of function? Because it's a question that I get from families, and for me, even I don't have the clear answer about this.
Yeah, probably Gabby can.
So no, it's a really important question, and one we get all of the time, because we know that it makes a lot of difference for the kind of treatments we go for with our children. It is a very intensive process to do it. It has to be done in a lab, and it requires a lot of intensive lab work to do the tests to determine if a mutation is gain or loss of function. And some of the mutations have been tested. It's called functional testing, one thing that it's called. And some of that has been done on some mutations, but not on a lot of them. There is a study that's happening now in a lab of a guy called Al George in Chicago, and he has funding to do this work with a number of different genetic treatments, including SCN8A. We're working with him now to help him understand the critical work that needs to be done here for SCN8A. So it's happening, but it's slow. Cinzia can also talk a little bit about it. She is funding a study in Italy to have some work done there to do more of this work. So it's a great question. Yes. Unfortunately, there's not an easy answer to it. But Cinzia, I'll give you the floor in a second. But the other thing that's happening is that we are learning, and we're trying through the diagnosis and treatment guidelines to also identify if there are some clinical features that we can look at that will help us understand and make an educated guess about whether a mutation is loss or gain of function. For example, we are pretty sure that if a kid does not do well on levetiracetam or Keppra, that's a pretty good sign that they have a gain of function mutation and they should be on a sodium channel blocker.
Okay.
So it's emerging. It's going to take more time. I encourage any family who is not sure about whether they have a gain or loss of function to join one of our family network meetings where we meet with Michael and talk about what we're learning from the registry, the different, we've gone through all of the literature and we can tell you which mutations have been functionally tested. So I'll leave it there. I don't know, Cinzia, if you wanted to say anything further.
No, Gabby, you explained everything very, very clear, so nothing to add. Thank you.
Yeah, and we'll invite Cinzia and her team to come in, and I'm sure she'll have them talk with the community more when they have more emerging results, but it just sadly is going to take time.
Yeah, it takes a lot of time. Yeah.
Yeah.
Is there,
Laura put in the chat, so neuros just assume what function a child has due to characteristics. I don't really know, unfortunately. We don't know what most neuros do, but that's why we also really are working hard to educate families so that they can bring the most emerging knowledge to their clinicians if their clinicians are not on top of the research. But I think hopefully what neuros are doing is checking the literature to see if this mutation is documented to have any functional testing done. And then if not, then they are hopefully looking at some of the characteristics like understanding if a kid does well on levetiracetam or Keppra or not, and then making an educated guess. So, but that's part of what we're trying to also figure out with the diagnosis and treatment guidelines is asking clinicians, do you look at whether a mutation is gain of function or loss of function? And if you do, what do you do with that information? And if you don't have that information, how do you decide how to clinically treat a child or adult?
Okay. Okay.
What would happen if both of those genes both areas were mutated? Has that happened? Where one side is not only mutated, but both?
I don't think there's any, in terms of a gain of function mutation, I don't think there's been a reported case of like a patient with 2 gain of function mutations on the 2 different copies. So we don't know, or that we know of, you know.
And in terms of loss of function, Miriam Meisler, who is here, I believe on the call, who was the discoverer of this gene, made a mouse that has 2 loss of function alleles, and that was, I think it was lethal early, at a few weeks or something like that, right? So probably you wouldn't see that in a person either.
Okay, thank you.
I have another question that is linked, that is a bit more technical, but I know that Google is providing a tool that can, from a gene, you can see what would be the shape of the protein. Is it something that as a searcher you can use to try to guess if it's going to be a gain of function or loss of function? Or is it totally utopic to think of something like that?
Are you talking about like the AlphaFold?
Yes.
So this advance in computational, sort of deep learning for protein function.
Yeah, exactly.
So that's very exciting. I don't think it would have immediate applications in this field. It's because we actually, the structures of sodium channels have been solved experimentally, so we sort of already know. And I think, but it might, they might be focusing on that problem of predicting mutations. But then the change in structure doesn't completely give you the change in function. I think, it would be a little tricky. So with the gain and loss of function, I just wanted to say, so my impression, Gabby probably knows this better, but is that gain of function is more common than loss of function in this community. Is it like 90% gain of function, something like that?
Yeah, so we, and I think this is probably true across any genetic mutation where there is a gain and loss of function component. We're learning is that there's probably a much larger population of loss of function, but they are slower to be able to get access to genetic testing because their issues might be less emergent. They're not having, tonic-clonics and showing real signs of a health condition. They might have, signs of autism spectrum disorder or delayed speech. So we believe that there is a much larger population of loss of function, people living with loss of function out there. And as we get better across the world doing genetic testing for people who have a range of different presentations and challenges, we think that there'll be a lot more. But you're right, about now it's 90-10, is what we think. And we're learning a ton from our loss of function families who are sharing their experiences. That is really helping us better understand the difference between loss and gain of function. And frankly, from the registry as well, because Michael and his team, Michael Hammer and his team are working actually on a really exciting new predictive tool that is taking what we're learning from the registry, things like age of onset, type of seizures, those kinds of things, and looking at predicting whether a mutation is gain or loss of function based on clinical features. So that is an emerging tool that as soon as it is, the kinks are all figured out, Michael is going to make available so that clinicians, if they're not sure, can kind of run this kind of test and see, you know, and have a best possible, and it's a very, very accurate as of right now. So, you know, there's a real chance that this could be something to help with clinical treatment of SCN8A before we get all of the functional testing done. So
there's a question in the chat on the specific variant may be one of both loss of function or gain of function, or can it only be either gain or loss of function? So there's an overall effect of a mutation as to whether it increases the activity of the channel or not. But within the experimental, you can look at lots of different features. There's like 12 or 15 features of sodium channel function, and there can be effects in kind of some that are more gain, some that are more loss, but then there's kind of like an overall effect that it will have.
So this is amazing discussion. I wonder if maybe we should keep going through the slides.
Yeah.
I think some of your upcoming slides are going to answer some questions people have. Feel free to keep dropping your questions in the chat, and we'll continue discussing as we go.
Yeah. Okay, so in terms of drugs, right, just, you know, most of us know this, but in terms of gain of function, you know, we have various AEDs that reduce sodium channel activity. There are some new NaV 1.6 specific blockers, which, you know, are exciting. And then, you know, we are also, our kids are on drugs that, you know, enhance the inhibitory activity of the brain to kind of, you know, reduce seizure activity. And then for loss of function right now, in terms of like, you know, targeting specifically this mutation and really having a treatment that is specific to a loss of function mutation, you know, there's not really an easy way to have a drug that can restore the activity, restore this mutated copy to being functional again. That's, you know, harder to think of something, a drug that could do that. And so I think most of the treatments that are being used are targeting the other symptoms that, you know, patients would have. So then in terms of genetic treatments, what's important to keep in mind with SCN8A, and, you know, all of this work here I'm showing is all from, and we've learned so much from Miriam's work, so is that, you know, this SCN8A is one of these Goldilocks genes, it's called. And so a Goldilocks gene means that, you know, if there's too much of it or if there's too little of it, that's not good, right? You need to have just the right amount to be able to have a healthy functioning brain. So again, mouse studies have shown that if, you know, you have 2 bad copies of SCN8A, or a complete knockout is what it's called, the mice will die within 3 weeks. And so, you know, this is not something that is viable. Again, we haven't, I don't think it's not been observed in people. You know, so again, if you don't have SCN8A at all, that's not good. But we also know that if you only have 1 copy of SCN8A, then you have the loss of function phenotype. And these are some of the phenotypes from the SCN8 Alliance data collection and the nice graphics that were posted recently. And some of the main symptoms are delayed speech, intellectual disability, developmental delays, autism. All right. And so, you know, there are different symptoms associated with having only 1 copy of SCN8A. On the other side, you know, we know that in our kids that have gain of function mutations, you know, there's seizures, hypotonia, intellectual disability, delays, movement disorders, you know, lots of different symptoms with that. And again, as kind of back to Tammy's question that, you know, I don't think there's any individuals that are known that have 2 gain of function copies. So that has not been observed. But then in between here, you know, is there a, you know, how much of a mutated copy do you need to actually have effects? And this is, you know, beautiful work from Mira Meisler's lab, you know, where she was able to create a model where you could a mouse model where you could kind of titrate up the amount of the mutant copy that was present and see like when is there enough of the mutated gene to cause seizures. And she showed that just a small amount of about 10% is enough to at least cause spontaneous seizures. So you don't need that much that is mutated to have an effect on seizures. So we have this kind of situation where, there's this kind of window in the middle where, there are no symptoms, the brain is healthy and functioning like it should be. But if you have too much or if you have too little, then, there are lots of different symptoms and features that come up, right? And so it means that, when you're trying to manipulate, the amount of that is present of SCN8A, you really have to be able to do so in a very finely tuned and controlled way, right? So that, if you have too much and you try to bring it down, that you don't overshoot. And so this is, an issue in terms of targeting these genes that are so-called these Goldilocks genes, and that make it more challenging. So then in terms of therapeutic options, so I just wanted to talk about gene therapy before we then get to the ASOs, because I think this is kind of what commonly, people hear about is gene therapy. And so currently for gain of function mutations, there's not really a gene therapy that is suitable, because most of the gene therapies that we have for diseases right now are to add a DNA sequence to make a protein that is not being made. And so, because our kids actually have too much SCN8A, right, we don't want to give more, right? So being able to, put in and add a new SCN8A gene is not going to be helpful. So that's not really a therapeutic option for gain of function. What could be down the line an option is actually correcting the DNA of the mutation so that, and we now have this CRISPR technology, which was discovered recently, where we are able to correct DNA. But, that is starting to be used in humans in different applications. But, using that to repair the brain is still a bit far off. It's hard to know, like, when this would be suitable, an option for repairing the brain, but it's not something that would be happening, anytime really soon, but it's definitely something that, people are working on to try and use CRISPR to repair. But so that's kind of what would be a gene, therapy option for gain of function mutations. Again, not kind of the most commonly used gene therapy to kind of add DNA, to replace or to kind of add something that's there. That's not a viable therapeutic option. It is a viable therapeutic option for loss of function mutations to be able to, be adding SCN8A DNA to make another functional copy of SCN8A. And again, that's kind of what this gene therapy for spinal muscular atrophy, Zolgensma, does, is it provides, more DNA to make a functional copy of a protein that's missing in SMA. So, these are kind of different approaches. Okay. So now we get to ASOs. And, ASOs are a really exciting new therapeutic avenue because they can do lots of things because they target the RNA. And so there are kind of two types of ASOs, and so we'll focus on the first type first. And basically what an ASO does, it's a little bit of RNA sequence that can associate with an mRNA in a very targeted way. So you can design it so that it only binds to a specific RNA sequence, and it will cause the degradation of that RNA so that the protein is not being made. So you're not touching the DNA sequence, doesn't matter. You just, once the mRNA is made, the ASO will bind to it and prevent it from becoming protein. So the first ASO for SCN8A was made by Miriam and her lab. And they made an ASO that degrades SCN8A mRNA. It's not specific to a mutation. It targets both copies. So it will degrade both the healthy and the mutated. And they published this wonderful paper last year. And I know there's a video also that the SCN8 Alliance made with one of the authors, Sophie, on this, breaking it down. So, this was really, really exciting that, an ASO could be used to reduce seizures and extend lifespan in mice that have mutations in SCN8A and that have this gain of function phenotype. So, this was like really, really exciting for our community that like this, that her lab was able to show this, because it really opens lots of doors for new treatment options. The potential risk, though, with this therapy is that if it works too well, right, we could reduce the amount of protein too much and turn into a loss of function phenotype, right? Because we have this Goldilocks level, if we get too much, then we could kind of, remove, like remove too much protein and have the loss of function symptoms. And so therapeutically, that can be a little, can be a bit scary in terms of like pharmaceutical companies or clinicians in terms of like giving a treatment like this, that like if it works too well. And again, remember, these ASOs will spend about 2 to 3 months in the blood, right? They stay in the body, they'll stay for quite a while. If you give too much and then you start to shift the phenotype to the other side, that could be a concern. So the approach that nLorem is taking is that what if we could then make ASOs that instead of targeting both copies of SCN8A and the mRNA, they actually make ASOs that are specific to a mutation. So you have an ASO that binds to a specific mutation in SCN8A, and so you make less of the mutated copy, but you don't have an effect on the healthy copy. And so this is the approach that's being used by nLorem for certain diseases that are caused by a gain of function, where what you're trying to do is reduce the protein, make less of it because it's too active, where they could design ASOs that are specific to a mutation and will just lead to the degradation of the mutated protein without touching the healthy one.
And so, yeah, so just to clarify that they are developing, we'll talk about nLorem in a moment, but they haven't actually tried this for SCN8A yet. This would just be hypothetically what an allele-specific ASO would do, one that targets just a mutated form. But that is their general approach for treating these rare diseases caused by gain-of-function mutations. Next. So what is the nLorem Foundation? So it's a recently created nonprofit foundation that was founded by Stan Crook, who was the CEO of this company Ionis Pharmaceuticals in San Diego that actually has been developing ASOs for decades now, including the Spinraza ASO for SMA that was mentioned earlier. And so their mission is to provide personalized experimental ASO medicines to treat what they call nano-rare patients, which we'll explain in a moment, for free for life. And they're now, they've accepted about 70 patients where they're now developing custom ASOs or attempting to develop them. There's no guarantee that they will be able to take their developmental pathway all the way to a successful ASO for any individual patient. Next. And these slides, I should emphasize, they were shared with us by Amy Williford from the nLorem Foundation. So what are nano-rare patients? So these are patients that have a mutation in a gene that is a very rare mutation. Either they're the only one that has that mutation, or there's estimated to be 30 or fewer people in the world that have that exact mutation. And so this would apply to virtually all SCN8A patients, actually, and patients with many other of these rare diseases. That is, there might be thousands of people that have a mutation in a particular gene, but they generally will be distributed across the gene, and so any individual mutation is extremely rare. And so the reason for focusing a nonprofit on this type of mutation is that there isn't really a viable commercial model for developing a drug that treats 30 or fewer patients. It's just the clinical trials would be impossible to run, and you couldn't like recoup the expenses that the research would cost. And so therefore, it needs to be thought of as something that could be done only on a nonprofit basis. And so we know about these nano-rare patients, and that they're, the diagnosis is a problem, and they're not good treatment options for many of these diseases. Next. So this is the sort of pipeline by which nLorem works. They first, the family of the patient works with their physician to apply to nLorem, and then the application that describes all the clinical and genetic information available for that patient is then evaluated by a board that kind of assesses whether this kind of custom ASO approach might be feasible for treating this particular gene, this particular disease, this particular patient. And then once you're accepted, and our daughter was recently accepted, then nLorem will develop a strategy for screening a bunch of ASOs. You can design ASOs to target a particular gene, knowing the sequence of that gene and a particular mutation, but they don't all work as predicted, and some may have side effects. And so you, in practice, need to screen hundreds of different, slightly different sequences to find the one that works best, and ideally doesn't have side effects or toxicity. And so to date, it's been about 70 patients, and most of those are currently in screening. For our daughter, they haven't started the screening yet. And once they find a sort of a lead ASO that works well in patient cells, ideally, and or other similar cells, and is not toxic in a mouse, for example, they will go into production of that ASO, and presumably make a lifetime supply of that ASO for the patient. And then they would apply to the FDA, submit an application to the FDA to be given just to this one patient. And right now they have about 5 at that stage, and they're hoping to begin treatment of the first few patients this fall. And just to clarify that these, the 70 patients they have, I think, span many, many different genes and diseases. It's roughly 70 different genes and diseases that they're kind of casting a wide net here. Next. So the idea of nLorem is to basically adopt the strategy for commercializing ASOs that this company Ionis uses, and basically use the screening capacity of Ionis and their production capacity, and sort of everything they know about developing ASOs, kind of apply that. And recently, because they've scaled up, nLorem now has their own labs as well that use for their screening activities. But they work very closely with Ionis and with existing pharmaceutical production and development companies. Next. Yeah, so we talked before about this version 1 of ASO, where you try to knock down or reduce the amount of mRNA and therefore reduce the amount of protein. And this is attractive for gain of function diseases. Of course, if the ASO works perfectly and degrades all of the mutated mRNA, then you would have only half of the normal dose of SCN8A in this case, and that would presumably trigger loss of function phenotypes. And so that's definitely a risk with this approach. Next. So it turns out there's another type of ASO that's been developed, which are called splice switching ASOs. So it has a different chemistry of the antisense oligonucleotide. And so to talk about how they work, we need to talk about a little bit more about another feature of the central dogma, which is sort of an added complexity that's very common in human genes, which is called alternative splicing. And so this is a process where from a single gene copy in the DNA, you make different RNAs. You first make a copy of the DNA, an RNA copy of the DNA called a pmRNA, and then that undergoes a process called splicing or alternative splicing, in which you produce different mRNAs. And so in this case, that turns out that human genes are broken into sort of alternating segments called exons and introns. The exons form part of the mature mRNA that will eventually direct the synthesis of a protein. And the introns are non-coding pieces that are in the RNA that's copied from the DNA, but then are removed at the RNA level and degraded. And so only the exons remain and then are ligated or stitched together. Next. And so in the case of SCN8A, there are about 28 exons or so, and most of them are present in all copies of the mRNA, that's these gray exons. But there is this region where these two alternative exons shown here in blue and red, where each mRNA either includes the blue exon or the red exon, producing two slightly different versions of the NAV 1.6 protein. Next. So the idea of splice switching ASO would be to design an ASO that targets one of these two exons. For example, if the mutation occurred in one of these alternative exons, as it does in Margot's case, then you could design the ASO to target the pre-mRNA, that is the RNA copy of the DNA that is made and before it's spliced. And if you could block the splicing machinery from recognizing that blue exon, then you wouldn't produce the blue mRNA or the blue protein. Instead, you would only produce the red mRNA next and red protein, which might be normal, for example. And so the general idea of splice switching ASOs is that when you have a gene that produces different mRNAs with different function, you can often design an ASO that will, and one of those is desirable and one is undesirable. You can design an ASO that will interfere in the splicing process and switch from the undesirable to the more desirable form without actually changing the level of expression of the gene.
So, so this is an approach that's, been used. And so this Spinraza treatment for spinal muscular atrophy, that is a ASO, it actually works in this splice switching way. So here's a little video that was made by Cold Spring Harbor, which is a research lab on Long Island where this technology was first discovered and then made into an ASO and a treatment for SMA that is used in patients. And this is a video that I think explains kind of this really well on how it works. So we can just watch this as an example.
While RNA editing is critical for generating the hundreds of thousands of proteins that keep us alive, splicing errors can lead to devastating health problems. One example is an often fatal disease called spinal muscular atrophy, or SMA. It's the #1 genetic cause of infant death. SMA is caused by too little of a vital protein called survival of motor neuron, or SMN, in the spinal cord's motor neurons. Too little of this protein causes the motor neurons to die and the muscles they control to waste away. The SMN1 gene, which produces the SMN protein, is missing or damaged beyond repair in SMA patients. Humans have a second SMN-producing gene called SMN2, but this gene is a poor backup because it produces very little functional SMN protein. This deficiency stems from a mistake during RNA editing. Remember, that's the process of forming a coherent RNA message by removing the gibberish letters or introns. But what is this mistake, and how does it cause SMA? Let's say the SMN gene generates RNA copies made up of introns and exons like this. Normal RNA splicing would delete the introns, the gibberish letters in blue, and leave only the exons, the red letters, generating the following message: draw a big house on the corner of the wide street. In SMA, it turns out that during RNA splicing, the 7th exon of SMN2 is skipped by the machinery that edits RNA. Skipping of exon 7 generates a faulty message: draw a big house on the wide street. Just like removing word number 7 in the sentence leads to a statement that makes no sense, removing exon 7 in the RNA results in a defective protein. Remember that SMA patients have to rely solely on the SMN2 gene to produce the survival of motor neuron protein. But due to exon skipping, not enough usable protein is made to support normal muscle development. Infants born with the most severe form of SMA often don't make it to their 2nd birthday. But researchers have been studying how to fix this splicing problem. One solution is called nusinersen. It enables SMA patients to make more SMN protein. But how? Nusinersen is a short sequence of RNA that binds just after exon 7 in the unedited RNA message. This tiny piece of RNA prevents exon 7 from being skipped. It is called an antisense oligonucleotide, or ASO. Exon 7 is now included in the edited RNA message, which in turn generates full-length functional SMN protein. By preventing exon 7 from being skipped, nusinersen reverses symptoms in SMA patients.
Okay, so I hope this was helpful in giving an example of how these ASOs can be used to then affect how much protein is being made. And, the fact that this is approved for, infants right when they're born, is really helpful for, trying to use these ASOs for other diseases, because we know that this method, we can inject these in the spinal cord, they can get to some neurons. And I wanted to bring this up too, because there's currently a clinical trial for Dravet syndrome for SCN8A that leverages this approach. And so, this is kind of exciting because they're recruiting patients. This is, different than what we would want to do for SCN8A, but it's just showing that, there is kind of exciting progress in this area where, there's using ASOs to treat sodium channel diseases. And so we'll also be able to learn things from that. So as just a brief background on Dravet, which is driven by mutations in SCN8A, which is a different gene. And it's different also because SCN8A is mostly present on these inhibitory neurons that kind of shut things down in the brain. So this is different than SCN8A. And most mutations in SCN8A cause a loss of function phenotype, where you only have, again, one good copy. So you don't have enough functional protein, this NAV 1.1 that is made from SCN8A. So you don't have enough inhibitory activity, and so you have too much positive excitatory activity that leads to seizures. And so this company Stoke Therapeutics that is developing ASOs, in the epilepsy space, is kind of using a similar approach. This is a bit complex, and I don't want to get into the nitty gritty, but this is from their website. And so, in a patient that has a mutation in a non-functional copy of SCN8A, which is still similar to our loss of function patients, right? So you have one copy that works well, where you get the protein that's made, it's normal, it's working. And then you have, this mutation that means that you have no functional protein being made. And so,
I can do this part. So a wrinkle in the expression of the SCN8A gene is that normally, even from a healthy copy of the gene, you produce both functional mRNA and this non-productive mRNA when you include this blue exon there. It's just a regulatory mechanism to control the level of SCN8A. And so the Stoke Therapeutics has figured out a way of kind of leveraging this in these loss of function Dravet patients, where they have this ASO, which is shown here in red. They call it Tango ASO, that blocks the inclusion of this blue exon. And remember, the blue exon is what makes the mRNA non-functional. And so then they will shift the splicing so that you produce more functional mRNA from the healthy copy of the gene, and therefore restore the levels of SCN8A or NAV 1.1 protein. And it's in phase 2 clinical trials currently. Next. Yeah, so in the SCN8A, as I mentioned before, there's two different versions of this exon 5, and the blue and the red, that produce these two different versions of the protein, which have similar overall activity, and that's controlled during development. Next. And so in Margot's case, we know that her mutation is found actually in one of these alternative exons, in this blue alternative exon, which is expressed earlier in development. And so the strategy that nLorem is proposing to pursue for treatment of Margot would be to develop an ASO that targets this blue exon. It wouldn't necessarily be specific for the mutant allele, like the mutant copy, but it would shift splicing away from the blue exon and therefore toward the red exon, which so you would make less of this mutant protein and more of the corrected of the adult protein isoform, which is normal in her, not mutated. And so this nLorem N of 1 ASO strategy is very new. They started taking applications in 2020. They have yet to treat their first patient. It's an experimental treatment. It comes with risks because it's never been tried before. Your kid will be the first one to ever get this ASO that hasn't been given to a person before. And there's also, even if they accept a patient into their pipeline, there's no guarantee that they will be able to develop an ASO that is effective, that does what they want it to do, and that is not toxic. So some of the patients have dropped out because the pipeline did not produce an ASO that they thought would be effective, and so it just doesn't. It's not guaranteed to succeed. But assuming that nLorem is able to develop a splice switching ASO therapy for Margot, and it's approved, then it will be, and that they estimate that takes probably 1 to 2 years of development. And if it is given to her, then it will be interesting to see whether SCN8A can be safely and effectively targeted in humans with an ASO. As Miriam has shown, it can be targeted in mouse models. And then also to see what effects it has on Margot's symptoms, on whether it can control her seizures, and whether it can aid her development. And Miriam has done some recent work suggesting that reversing the sort of using a treatment later, even after developmental delay has occurred, some of it may actually be reversible, which is very helpful.
So back to Tammy's question about, right now this is being made in terms of the mutations. So Margot's, this region that switches, that has the 2 versions, is between amino acids 206 and 236. So if patients have mutations in this range, like potentially this amino acid, this, this ASO could help induce that switch for any patient. It potentially, so it's, and there are actually about, in the registry, about 30 patients that have reported mutations in this region. So, so there's that. There's also some experimental evidence that, if you do switch this, the adult part is actually less active than the neonatal one. So this could be a way to kind of tone down the activity of NAV 1.6 in a very specific way. And so this is actually what we're looking at in my lab. So, nLorem is developing the clinical aspect, but in my lab, I've now hired a PhD student to work on this and to understand, if we can switch this, does it impact, can we reduce the activity of NAV 1.6 in, in mutations in this region and then in mutations in other part of the gene. So that's kind of what my lab is looking at, to see if this could then be expanded, because we know that inducing this switch makes it less active. So this could be an attractive way to reduce the activity in a specific way. So those are, the questions that were research questions we're looking into.
Yeah, and I just wanted to comment here that the amino acids 206 to 236 can be encoded either by the 5N neonatal exon or by the 5A adult exon. And so there are patients with mutations in each of these 2 exons. So if switching from 5N to 5A would only be useful if the mutation is in the 5N exon. And that is not always super clear from the genetic testing report. Basically, because the amino acid numbers, the 2 exons code for the same block of amino acids. And so the amino acid number doesn't actually tell you which exon it's in. You have to look at the chromosomal location and check where that exon is. And so that's just but for patients with mutations in the 5A exon, then they might be amenable to this approach in reverse, to develop a different ASO that would block the 5A exon.
And yeah, there is a lot of kind of data from Al George from Northwestern that Gabby mentioned, where he's looking at how this different switch affects the mutation, the activity of sodium channels with other mutations. So that's something that's being looked at as well. And then I think there was a question from Shinsia: is there a maximum age at which it is still possible to reverse the disease? Yeah, I have no idea. I mean, I don't know if Miriam wants to comment. She's doing some things on that in terms of reversing. What's the maximum age that you can reverse the disease? I don't know if you want to comment, Mary.
We haven't quite asked it that way. We've
Yeah.
asked it early in the disease, but it's a good question. We should look at that.
So I know for SCN8A, there was a paper that came out recently where they, also kind of reversed the SCN8A.
Madeleine, are you there? I think Madeleine is having a technical issue, but she's working on it, and I'm sure she will rejoin us shortly. If there are any questions related, I'd be happy to take any questions if there are.
So there was a question Zipporah had. I don't know if you want to ask it, Zipporah, or if you want me to just read it about mitochondria.
And I think I asked this before, but I can't remember. So Mosiah, there is a mitochondrial mutation that they found. And so I was trying to figure out if that possibly could play a role because the mutation is on the exon 17 or 19 or something like that. I can't remember exactly. But any significance, any connection?
So you said it's a mutation in a mitochondrial gene. So it's a different gene. So not SCN8A. Is that?
No, but I didn't know. I mean, I don't know. So I'm just asking, I don't know how it's connected, if it possibly could be connected.
Yeah, I mean, I don't know. There's some, mitochondria affect metabolism, and there are connections between metabolism and epilepsy and the brain and so forth. For example, keto diet kind of changes metabolism and is therapeutic for some kids. So I guess maybe there might be a dietary intervention if there's a mitochondrial issue, but yeah, I think we should.
We don't know.
We don't know. We're not
We don't
medically trained. So
Depends.
we shouldn't comment on that.
I think in general, though, there is there's emerging thinking that it's very likely that if there are other mutations that somebody has, whether they are related to epilepsy or not, they could somehow impact the SCN8A mutation, either helping or hurting based on whatever the function of that secondary mutation is. So it's likely that there could be an impact from it, but I think it's very early for us to know if that's the case or not. So
You answered exactly what I was trying to ask. Thank you.
yeah.
Yeah, and Miriam has another very nice paper where she's shown an example of a gene that when it's that can impact how severe the phenotype is. I mean, and that's just one gene. So yeah, there is, she has shown that can happen. So, but we don't know how many genes can do that, or which ones are all the ones that do or don't. So a question from Chintzia, my son has our gene 1872 plus CACNA1 mutation. Could it be possible candidate? I mean, they decide, so they will look and, the clinicians they submit the application, and they have a committee with scientists and ethical people who work in ethics. And, there's lots of kind of parameters that go into it. We have no idea. Like, it's not, we don't, they have a process by which they decide.
I think one other factor that might be is that 1872 is one of the most common variants within SCN8A. So there's over 30. I don't know how hard that line is for up to having only 30 people in the world or whatever.
I would just go ahead and apply and just see. Yeah, again,
it doesn't hurt.
May not be an absolute requirement, or they can tell they'll you consult with your neurologist and submit the application, and I think there can be a little back and forth with them or something like that. But yeah.
Maybe, and people can feel free to just jump in and ask questions, but I was wondering maybe if you guys could talk a little bit about your decision to, explore nLorem as an option. I mean, you shared a lot about how it works and why it works, but, and we have potential treatments on the horizon with Neurocrine and Praxis, and Margot has this kind of very specific, was it this very specific, kind of intron, I'm gonna get the words wrong, but like this,
This alternative exon,
Yeah, the
alternative
splicing.
exon,
yeah.
that really made you guys think, she is so unique and it might be worth trying that. Maybe just a little.
I mean, I think we're generally, we know what the current medications can do, which is they can partially control the seizures, but not not restore her development, or we know where what the current therapies can do, which is not everything we'd like, of course. And so we're very interested and open to new approaches. So we're excited about the Neurocrine trial. We're excited about the Praxis trial. We may enroll Margot in one of those. I mean, I don't think these things are mutually exclusive, and we don't know which ones are going to work ultimately. So we want to learn as much as we can, and we're open to trying things there. I mean, I like the general drugs that Madeleine is mentioning. The sodium channel blockers, they block many or all of your sodium channels, and some of those are in the inhibitory pathway. So they're kind of a blunt instrument, and so something like the Neurocrine drug, which just targets NAP 1.6, that's exciting. We like being more precise, more and that's also one reason to like the ASO therapies, is they're just treating that gene and potentially even that mutation, or at least the exon that has the mutation. So it's that they're more specific and targeted. They're further upstream in the pathogenic pathway, and therefore you might hope that they would more completely block the pathogenesis. And so.
I mean, it is scary because it's like a new treatment, and it would be, if we get to that point that there is something that is ready to be injected, obviously that's, there's
going to be that moment,
scary.
and we're not going to we're going to think it'll be very real, when we get closer to it and we see what it happened in cells and what happens in a mouse, and but
I think it's, I don't know, as scientists, it's like really exciting to think that these treatments are available, and we want to help push the science forward too, in the most in the safest way, the most we can learn, and I think this idea too that we can kind of correct the protein, like what effects is that going to have? Because I think for SCN8A in humans, we don't know that, and I think there's so much to learn from that could then spur the development of other genetic type treatments. So I think that's more exciting than anything. And I think
and also the fact that this Spinraza drug, it really works well in SMA was a disease that had no treatments, and kids often died very, very young, age 2 or in later in childhood, and it was a progressive muscle degeneration disease, and then Spinraza was the first FDA-approved drug for this, and it often halted the muscle degeneration, sometimes even improved muscle function. So it's very very exciting. Has some side effects. These injections are not fun, and can cause some toxicity. But overall, it has been very successful, and that bodes well. And there are other ASO drugs. It's not the only one. There's a dozen or 5 to 10 other ASO drugs. Some work by splice switching, some work by this knockdown strategy. So it is a viable therapeutic approach. And so I think the Spinraza is really what got everyone's attention. That it got like various prizes and things like that because it was the first. So. Yeah,
Yeah, and I think it's, this is just the beginning, right? I mean, this is very early days. SCN8A and so many of these disorders, genetic disorders, were discovered just 10 years ago, right? So we're still really just figuring it out. And I think the fact that we've got things like nLorem, we've got, you know, really committed scientists like Miriam. For anybody who doesn't know, Miriam Meisler has been studying SCN8A since 1990, I think you said it was, right, Miriam? We tried to count back the years, something like that. So she's been studying SCN8A before we even knew that it caused epilepsy in humans. She always thought it probably did, and she was waiting for Michael's call, and then it finally happened. And then we know that there are other scientists that are exploring CRISPR type treatments for SCN8A, but there's a long way to go and a lot to learn. But it's really an exciting time. So I know that we've got Miriam here who's a researcher. I know I saw Roberto. Is he still on? I mean, if he dropped off. He's a researcher in Italy, a scientist and clinician. And I love that we've got grandparents represented. We've got a couple pairs and Jayetta. I don't know if there's any other grandparents on, but if you guys have questions too, feel free to jump in. Any families, or you guys might have already got the whole rundown. You probably were the test case for hearing all of the science with Madeleine and Chris.
Yeah, I see Tammy's question there, and I think it's hard to know, Tammy, whether other, you know, other genes that are mutated could have effects. Like it's really hard to know. Like just by knowing them, like you have to look experimentally. So it's impossible to know at this point.
And they could be protective in some way too. We don't know if it's making things worse or better.
Right, exactly. Yeah. It's
Thank
you
you.
know,
yeah, because we do
you
know
that's
of
probably
cases,
what's
yeah.
you know, I mean, when we heard, you know, there's, you know, some patients that inherit their mutations, you know, from a parent, but the parent's unaffected and then the child is affected, you know, and that's probably other genes coming into play that are, you know, leading to the fact that the parent had no seizures versus the child does, you know, and
the microarray is inherited. And
okay, yeah. So yeah,
so,
it's so hard to know.
but the SCN8A is not. So you wonder if, you know, if like the microarray, because these are all neurological diseases, if maybe it's making him better or worse. You don't know.
For sure.
Yeah.
For sure.
Yeah.
And I think in 10 years we'll know a lot more.
Yeah, no, for sure. I mean, these gene interactions are not well studied. They're very hard to analyze because there's usually so few of any specific interaction, but probably those other genes are affecting, you know, like not all the 1872 kids have the same, you know, have the same symptoms. Some are worse, some are better, and maybe these other mutations in these other genes are influencing that, you know. So that's maybe that could be studied a little bit.
And that's something that Michael has been talking about for a long time. He's been talking about, okay, and he calls them like modifier genes, right? So could it be, you know, modifying what's happening with SCN8A from, you know, a positive standpoint or a negative standpoint? Because, you know, SCN8A is a sodium channel, but it doesn't function without the calcium channels and the potassium channels. They're all interconnected. And so like if somebody, you know, Chinzia, you mentioned that there was a calcium mutation, you know, the way that the sodium channel works is in coordination with the calcium channels. So if there's some kind of protective or, you know, kind of more challenging mutation there that could be impacting. So it's really, we have so much to learn, but frankly, that's kind of what Michael is doing with the registry too, right? So the data that he's gathering, he is looking, you know, when people send in their genetic reports, he's able to look and see, and a lot of the analysis that he's doing is saying, okay, is it SCN8A only, or is it SCN8A plus? Is there something else happening here that might be amplifying or modifying or deteriorating the impact of the SCN8A mutation? So it's really cutting edge stuff that's really exciting. And that's why, you know, we've got a registry drive coming up. We're really excited and hopeful that we can get a lot of participation from families, because that's the kind of stuff. And Michael doesn't just keep it to himself. This is data that he's sharing with any researcher that is interested in digging in and learning more. So your data from that, and he's also starting to analyze, as soon as we can get it, it's been kind of hard, but looking at data from Citizen in coordination with the registry. So that's interesting because we'll be able to finally start to link clinical data, right, directly from doctors and tests with what parents report, which each come with their own strengths and validity, right? I mean, doctors are not going to be able to speak to what we experience day in and day out at home. They just have no idea. So the registry helps capture that. Whereas, you know, testing done in the hospital with EEGs and MRIs and, you know, even just, you know, CBCs, you know, basic blood work can help reveal kind of some of these other things that might be going on with our kids.
Yeah, and Miriam also had another cool paper that came out recently where they used her SCN8A targeting ASOs in mouse models of other epilepsies. So for example, like this KCNQ2, right, which is a potassium channel, you know, another rare epilepsy. And they used the SCN8A targeting treatment and it had effects in the KCNQ2 driven model. So that's cool because that also kind of shows that, you know, developing treatments for SCN8A could not just help our kids, but also other mutations, other epilepsies, which can then, you know, help pharma be more excited and put more money into targeting SCN8A because it's such a powerful gene that, you know, this would help a larger population of patients. So that's also, you know, really exciting for those experiments from her lab, because that really will help, you know, invest more into treatments targeting SCN8A because they could help so many other populations. So thanks again, Miriam.
Yeah, and also in the original paper, wasn't there a Dravet mouse model too that was treated with the SCN8A ASO? So there's a lot of potential that you know, for having these better tools to target, you know, specific sodium channels could have lots of interesting applications. So it is. We were at this epilepsy Gordon conference, research conference in Barcelona last summer, and there was a good number of people working on SCN8A researchers, a half a dozen or so, and as well as Dravet was well represented. And so there's a lot of interest in the sodium channels in the scientific community, which makes us really, you know, optimistic that things will move quickly. You know, we're actually, although of course we're all unlucky to have these mutations, but we are also lucky that they're in a gene where there is an active scientific community that's working on it. That's not true of some other rare genetic diseases. And where we have a lot of very well organized community with the registry, with the alliance, with acute syndrome, etc. So.
Miriam, I want to give you an opportunity if you want to say anything. I know you've been in this space so long, you're continuing to do exciting work with the ASO to investigate further.
Well, you know, there's two things on my mind. One is, it's very exciting what Madeleine mentioned, that knocking down the splice blocking of exon 5N might also depress overall expression and then be relevant to all of these mutations. That's really exciting. The other thing that's really a dilemma is this Goldilocks aspect. And if we take a gain of function, and if we eliminate the mutant allele by an allele-specific treatment, then we've converted it to loss of function. And so I'm often asked this question, and I always say, well, I don't know, how would the parents feel about that? I mean, the loss of function phenotypes do seem much more mild than the gain of function.
Yeah, I think that
Would anybody really try to eliminate a gain of function allele with knowing that you get these other effects with loss of function?
Yeah, I think that came up at our last research meeting. We have a quarterly research meeting with researchers doing a lot of lab work, basic science work in this area. And that question came up, and it's one that I think would be interesting to put to the community, right? I mean, personally, as somebody who's got a very severely affected kid, my son Elliot is 9, for anyone who doesn't know, and he's he still hasn't met any of his developmental milestones. He has no head control. He can't talk or walk or sit on his own, can't eat by mouth. And to think that potentially we could have a kid who might be able to have some better control, be able to have some increased development. Of course, we don't know after many, many years of seizures and brain damage and, you know, whatever is going on, whether that will be able to be repaired. But I think personally, I would welcome having a kid who has, you know, less severe seizures. Likely most loss of function kids are having more kind of absence type seizures, right? That are not as damaging and could potentially have more engagement. I think most of us, you know, almost all kids with SCN8A have severe communication and speech delays. And if there's improvement in that space where a child can start communicating more fully their needs and their challenges, that's a really, really powerful way to be able to help improve your child's quality of life if they can tell you what's wrong or what they need. So anyone else have any questions or thoughts? Let's see, comment here. Zane, am I lost? You're welcome to come off mute if you want, Zane, or I can read.
Hi, yeah, so we have a 4-year-old son, and he also has this SCN8A genetic mutation, but he's definitely lost some function. He's been nonverbal, but he does, you know, we've never experienced him to have any seizures. It's mostly like comes off as like intellectual, kind of like the speech delay and more like intellectual delay. Yeah, so we were just kind of interested in learning more about that. But you know, he's been getting therapy since he was 18 months, and you know, speech, occupational ABA, and he's been making a lot of progress. So yeah. So I just kind of wanted to share that kind of experience.
That's wonderful. Yeah. And I should clarify, when I say I would gladly have kind of more loss of function features, that doesn't mean that it's not really also really challenging to have a kid who has loss of function. I mean, I'm sure, Zane, you guys have been through a lot of challenging times, and it's intensive, you know, work to, you know, have a kid in that many therapies and constantly getting pushed. But
yeah, I just kind of wanted to share my experience, as in, I mean, the gain of function definitely sounds a lot more severe. So if anybody was wanting to know what loss of function is like, yeah, I mean, there is delays, but thankfully we don't have, we haven't had any like seizure or epilepsy type, no physical disabilities. He's, you know, he's been able to, can run around. He's pretty physically strong. Yeah.
Oh, wonderful. That's terrific.
Yeah. Thank you.
Thanks for sharing, Zane. Anyone else? I see a couple of new names on here who we've never met before. Feel free to introduce yourself, share if you want, or ask any questions. It's really nice to be in community with all of you and talking about this and hearing about how your kids are doing.
Hey, Bobby.
Hey, Julia. Dylan, we'll come to you right next.
No, I was just going to say, obviously I'm watching, but I'm doing something else at the same time. I'm walking, but I just wanted to say that it's so clear. Like, so thank you for taking the time, Madeleine and Chris and Miriam and everyone, for explaining all of that. I mean, it's such a complicated topic, but it was so clear the way it was laid out. And it's just so exciting to see so much work being done in this field and with the ASO. And I'm just so excited to see the progress and see what happens over the next, you know, year or two that's being developed and what that means. So just wanted to say thank you. No questions, but it was an awesome webinar.
Yeah,
thanks, Julia.
And thanks for what you're doing, Julia. We know you guys are working hard to have some research done at Sick Kids, which we're really excited to see come together. So it's a lot of really amazing leaders in this space in the SCN8A community who are working. All right, Dylan, I know you were gonna maybe come off and say hi.
Yes, hi. Thank you very much for this talk, Christopher, Madeleine, and thank you, Gabi and Miriam for the wonderful work. I'm just a recent a newcomer to the SCN8A family. My family, we just recently figured out that my brother has a mutation, and well, we knew he had something for like years, but we never got a name. So now we finally have a name, and I'm trying to catch up with the nitty gritty of the details. I have always been puzzled by the fact that he was under control for 13 years on one medication, and then out of a sudden that completely changed. And I'm just wondering if that could be related with the change of this exon. Do we know about the time frame and changes of expression of these two, Chris, Madeleine?
Yeah.
I'm reading here, but is that
Yeah, I
anything
mean,
related with adolescence?
so we know, I mean, we know what how the two different splice variants changed during development. There was a recent paper last year about this, where they're about 50-50 at birth, and then over time the 5A goes up and the 5N goes down, and that's true in a mouse as well. But we don't really know how that impacts the clinical presentation, and there's not so many not so many examples with mutations in these two exons, and it's not really that clear. And then I think there's also Miriam's data showing that even a 10% dose of the mutant mRNA and protein can cause seizures. And so even if you would think that if you have a mutation in 5N, you would kind of have most severe symptoms early and they would get better. But when would they get better? It might not be until adulthood or something, and the 5A might be the reverse. But I don't think we have enough actual clinical reports to actually know if that's true or not. And it could be, it could be more complicated, and so yeah, it's unfortunately
Yeah, it's hard to know, but
really know. But you can look at if you know the location of the mutation, that should tell you whether which exon it's in, and if that might be relevant, I guess.
And the other, and Dylan, just one other thing that came up too is somebody mentioned is that potentially, I don't know if it could have been around puberty time. Puberty, we know, is a time of intense change in terms of hormones and the way things work. And that potentially could have shifted if that was around that time as well.
Yes, that was exactly the time. And I see that this channel is expressed not only in the brain, but also in endocrine tissue. So who knows what this thing is doing there. And yeah, lots of questions. But regarding that mouse model, is this change in the expression anywhere related with environmental factors, or it's just a developmental thing that is set in stone? One would imagine that the mouse model is in pristine condition to study that question.
In terms of the splicing, how the developmental change, whether it's purely deterministic or whether it's influenced by environment. Yeah, I mean, I don't think we know that. The mice that were studied were, as you probably in a in a very simple laboratory environment, but we know that stresses and infectious disease and other things can influence splicing, and so it's almost certainly influenced by the environment, and that's another factor that could be influencing also why some patients get more or less severe symptoms, I suppose. Yeah.
And one last question. I don't want to take anyone else's time, but just following with what Gabi was saying in terms of communication. Is there a way that I could improve how I communicate with my brother? Because he lost his speech. He used to have like 20, 30 words vocabulary, but all of a sudden after the seizure started, he lost that. But I'm sure there must be like techniques or charts or all the kinds of things that I could be applying to still have that communication. I don't know if he's in pain. I don't know if he's thirsty. I don't know if it's like all of those things.
Yeah. Yeah, I mean, I would work with therapists and like AC communication. I'm sure Gabby has some good resources too, and on her deep connections. And yeah, there's definitely lots of great ways to communicate with individuals who are nonverbal with AC devices and things like that. I mean, yeah, we're not experts in that at all, but so I know there are definitely resources for sure. But
yeah. Dylan, feel free to email me. I just put my email in the chat, and I can definitely send you some resources we've had some recent webinars that we've done with a different project we have on communication for those who are nonverbal. And so I can refer you to those. And I also want to take a chance to invite everybody to join us. We have weekly meetings with Michael Hammer, the geneticist who discovered SCN8A as causing epilepsy in his daughter, Shay, and therefore in humans. And we talk about the genetics, we talk about what's going on with our family members, and you get advice from other family members about things that have worked for them, challenges they're having. So I put a link in the chat as well about to join for all of our events. You can come to any meeting you want, even if it doesn't necessarily match where your child or family member is on the spectrum. It's an opportunity to just be in community with other people who are living with SCN8A and their families' lives. Hi, Liam. Time for the SCN8A kiddos to come and say hi. So yeah, the network meetings are a really incredible way to learn about what we're finding with SCN8A from the research, from the registry, and then also to share your challenges and questions and kind of try to, and we learn from you, and we're able to kind of help each other think about new resources to try or things like that. So we've got some folks on here who are regulars at our meetings. I don't know if anybody wants to say what those are. I mean, I speak about them and I'm a big fan, but you don't have to take it from me.
yeah, we love going and chatting.
What did you ask about, Gabby? He was screaming in my ear.
Liam, young man. Yeah. I was talking. Don't interrupt me, young man.
Oh yeah.
I'm just kidding. We're talking about the network meetings. Are they a useful space to, what do you think?
Oh yeah, I learned so much from them all the time. Like even the other day, there's somebody that's another child, maybe mutated, not in the same area, but kind of in the same, just things like that. So then I get to meet other parents that might be similar to him because we're the only one, mutated where we're at, and just stuff like that.
And you learn from people like Tammy. Tammy, if you don't know her, she knows everybody and everything. And she's incredibly smart and comes up with new theories about what's going on. And so you get the wisdom of other families as well who live this and breathe this and do a lot of troubleshooting. So it's
don't you start
really fantastic. So if you look, we've got a meeting this afternoon in 50 minutes if anybody wants to get on and keep talking. But unless there are other questions or comments, I don't want to cut anybody off. This has been amazing. Well, I'll just take a chance then to thank you guys, Chris and Madeleine. You guys, this is just incredible. I am so excited that we have this resource for families now to be able to reference and better understand. I mean, I've seen you give this talk already to me, and then I keep learning new things. So it's so complicated if you don't understand genetics and RNA and all of it. So thank you for sharing your science brains, your SCN8A parental wisdom, and bringing this to the community. I think we'll continue to try to do these. I'm thinking that one of our next sessions will be on family planning with SCN8A. If other people thinking about having other children, how do you consider that? So if there are topics that people would want to hear more about, Gina, it's so good to see you. I'm sorry we didn't get to hear from you. Did she drop? Yeah. Oh, there she is. If you want to say hi, Gina, you're welcome to come on. But so feel free to share topics that you'd like to have more discussion about, and we'd like to plan more discussions like this.
Yeah, no, thanks. We're really,
Yeah.
thank you all for coming. And we're really happy to be part of this community. And we really enjoy all our interactions. And,
Yeah.
and we can all help each other, I think, deal with this disease.
Dylan, we have a meeting in Spanish that we meet every 3 months. So when you email me, I'll send you that so that we want to make sure that all families have an opportunity to discuss and learn. So I'll share the link with you. So just send me that email. Thanks, everybody.
Amazing. Thank you.
Enjoy the rest of your
Bye.
weekend. Take care.
Bye.
Thank
you
you.
all.
Grazie.
Great.
Bye,
Ciao.
Margaret. Thank you. I will.
Bye. Great to see you, Chris and Madeline. And
Thank
Mary
you. Thanks a
Jo,
lot.
Karen.
Thank you. Bye,
Ciao,
Karen.
ciao, ciao.
Thank you. Bye.
Gabby, thanks for
Bye,
being here.
everybody. Great job.
Bye.